Hello, everyone. My name is Jake Russin. I work at the NIMH for Dr. Carolyn Beebe Smith in the section on neuroadaptation and protein metabolism. We study the rates of cerebral protein synthesis in certain animal models of disease, including one mouse model of Fragile X syndrome, which is a form of autism. And so for my project, I analyzed some of our results from the lab. So first I'll give some background on Fragile X syndrome and the method that we use to study rates of cerebral protein synthesis, which is called quantitative autoradiography. Then I'll talk a little bit about the goal that I had for the project in analyzing the data obtained from that method. And then I'll just show some visualizations of the data and how I cleaned it up and got it ready for what I ended up doing with it, which was to simulate some experiments in R in order to gain a better understanding of particular strategies that I should implement to obtain the best results in future experiments. So, Fragile X syndrome is, in humans, the most common inherited form of mental retardation. Symptoms include autistic behaviors, affective problems, varying degrees of cognitive impairment. It's a genetic disorder caused by the expansion of CGG repeats in the FMR1 gene, leading to the loss of the protein product, Fragile X mental retardation protein. The mouse model that we work with is a FMR1 knockout mouse, and it has been shown that this mouse has elevated rates of protein synthesis in certain areas of the brain. So collecting the data, the method is called quantitative autoradiography. Well, first some background. So proteins are synthesized by binding amino acids together according to the information in the mRNA. So what quantitative autoradiography does is that we label certain amino acids with radioactive tracer so that when we infuse the mouse with that radioactive tracer for a specific amount of time, we can later on tell how much protein was synthesized by looking at how much of that radioactive amino acid was incorporated into protein during that specific amount of time. So what we do is we infuse with the radioactive amino acid for a specific amount of time, then we dissect the brain and slice it, and we record how much radioactivity is present in those slices, and then we can calculate based on that amount of activity how much protein was synthesized during the time in which the mouse brain was infused with radioactive amino acid. So here on the right I have some figures. That the one on the top just shows the process of protein synthesis from amino acids, and the bottom one shows an example of what an autoradiograph looks like. So you have the color corresponding to how much activity is present in the cells at the end of the experiment, and then there's those are both mouse hippocampus. The top one is from a wild type mouse, and the bottom one is from a FMR1 knockout mouse. And so you can see that in general, because it looks like it's more red in general on the bottom autoradiograph, that indicates that the rate of protein synthesis in that mouse was a little bit higher. So the goal of this analysis was to let the data gathered by that method, the in vivo method, let that data inform the an in vitro implementation of the method. So we're setting up an in vitro method, and we want to know which area of the hippocampus would be best to sample from during that method in order to yield the best results. So in vivo is when you have cells that are in their normal biological environment, and in vitro is when you remove those cells from their normal biological environment and keep them in an artificial environment in order to study them more closely. So the main benefits of the in vitro method is speed and efficiency, and that's because we don't have to perform surgeries to implant catheters into the mice beforehand, and control because we have in the artificial environment direct access to the cells. So as I said, the goal of this analysis is to enable a best choice of the area of the hippocampus to sample from using that in vivo data. So the in vivo data comes in the following format. You have rates of cerebral protein synthesis values from 7 mice. 3 of those mice were wild types, 4 were FMR1 knockouts. And then for each brain, you have 150 to 200 approximately slices spanning the rostrocaudal axis. So the rostrocaudal axis is shown in the top figure on the right. The rostral means closer to the nose, caudal means closer to the tail, and slices are taken coronally, shown in B in that figure, and so there's about 150 to 200 spanning that axis. And then for each slice you have 2 values of RCPS, the value for the hippocampus in the left hemisphere and the value for the hippocampus in the right hemisphere. So overall you have 7 mice, each with 150 to 200 slices, and each slice has 2 measurements, one for left and one for right. So this figure just shows an example of what the data looks like from one hemisphere, from one mouse. X48 is the number given to the mouse, and this is the left hemisphere. So you have on the y-axis the rate of cerebral protein synthesis, and on the x-axis is shown the slice number corresponding to a particular spot on the rostrocaudal axis. So those first slices were closer to the most rostral portion of the hippocampus, and then the last ones were the most caudal. So you can see that the data is pretty noisy, and that there are some gaps in it, but overall the trend is that, and this is seen across samples, is that you have a decrease in the most rostral as you move caudally, and then a sharper increase at the most caudal section before a final decrease again. So in order to clean up the data, I did 2 main things. One was to fill all of the gaps by linear interpolation. That's shown on the right there, and then also each curve was smoothed using a moving average. Here, this figure just shows all of the data together. So we have one curve per hemisphere per mouse, so there's 14 curves in total, and you can see that they do agree in general on the basic profile, where it's a decrease followed by a sharp increase in that caudal portion. But it is noisy and highly variable. So to better visualize this, we have averaged across the genotypes. So that red line represents all of the Fragile X syndrome mice, and the black line represents all of the wild type mice, just averaged together. So you can see that the difference between the two in terms of rates of protein synthesis is pretty even across the entire rostrocaudal axis. So the question that I wanted to answer with this analysis is the following. Given the relationship between RCPS and the rostrocaudal axis, as shown in those previous figures, what area of the hippocampus would be best to sample from in order to get the most robust genotype effect? So the in vitro method is noisier and harder to control, so we want to be precise about which area of the hippocampus is going to allow us to see that difference in genotypes the best. So there are a number of possible slicing strategies, I call it, and each slicing strategy is defined by two parameters. There's the number of slices, so just the total volume of hippocampus sampled, and there's the particular spot along the rostrocaudal axis that volume of hippocampus is sampled from. So you can see some kind of examples of slicing strategies in that figure. So you can sample from the front of the brain, from the middle of the brain, from the back of the brain, and you can sample in different sizes. So the approach I've took to compare those different strategies was for every possible strategy to simulate many in vitro experiments in R using the in vivo data set as a kind of model for the population of Fragile X mice and wild type mice. And then compare the results from those different simulations based on which strategy produced the strongest difference between the genotypes. So some pseudocode just to illustrate how the simulations went. So I would randomly choose one of the mice depending on which genotype it came from. So if I want a wild type, I randomly choose one of the wild type profiles. If I want a fragile, I randomly choose one of the fragile profiles. Then noise was added to the target starting point. So whatever slicing strategy I was implementing has a designated starting point, but I wanted to add noise to that starting point so that sometimes I would start a little bit before, sometimes I would start a little bit after, and that's because in vitro it's very hard to control the exact starting point. You have to do it visually, basically. So in order to account for that in the simulation, I just add some noise, rostral or caudal, to the starting point. And then from the chosen mouse's data, I sample the area of hippocampus, the total volume designated by the particular strategy being implemented. And then I just sum across that area in order to obtain in the end just one final RCPS value for the whole hippocampus. And so we do lots of simulations for each strategy in order to produce a distribution, one for the wild type mice and one for the Fragile X mice, based on that particular strategy. So you get for each strategy one of these distributions of many, many, many simulations. And here you can just see, as is the case with most strategies, that the Fragile X data is in general higher on the RCPS than the wild type data, and that's just because the Fragiles in general had higher RCPS. So as I said, many simulations were done for each strategy, producing one of those distributions for each, and then these distributions were compared by calculating how great the difference between wild type and Fragile X was. and then the strategy chosen was just the one that produced the most robust difference between wild type and fragile. Yep, and that's what I did for my project. Thank you very much.